Hello everybody. Good afternoon. I'm Dr. Alauddin from Pakistan. The topic is isolation and structural elucidation of new cyclotetrapeptide Ochrine A and B from Aspergillus ochraceopetaliformis. I have completed this work in USA, University of California, Santa Cruz with Pelcrew, when in Mon, while the Frederick Willard is from Michigan, Henry Ford Cancer Center, while Maria Draskovic, microbiologist. Okay. So can I change it from here? Okay. Aspergillus ochraceopetaliformis is a marine fungus collected from Hawaiian shallow water sediment, and taxonomic identification were carried out at the University of Texas by these Branwicks and Thompson. Well, it is preserved in the glycerol stock here, while the strand is preserved in the liquid nitrogen in the glycerol stock at UCSC, University of California. But the question is that why marine fungi? Why marine? So it represents the 70% of the Earth's surface. Less than 1% of the microbial biodiversity is known, and we are looking for the unique defensive organic compound which may be due to evaluation and biodiversity. And as compared to terrestrial compound, we have very limited number of marine compound, and actually we are always in search for multi-drug resistance compound strand against the multi-drug resistance strand for new antibiotic and low toxicity compound. So it is just the introduction of my work. That why we have started. What's the prelude of our research work? If you look into the ziconotide, which is a harbinger, the first marine-derived drug approved by the FDA, which is 100 to 1,000 times more potent than morphine. So besides that one, marine fungi, a proven source of bioactive natural product. You can just look at that. From marine fungi, we have collected from different substrates, like look into that from fish and from crab shell and from others. So we have different type of compound like alkaloid, which have human leukemia selectivity, peptides, which have different type of cancer activity, polyketides, steroids, and other. And all these are all these were get isolated from marine derived fungi. So that was so another thing about the fungi. And you can play with fungi inside your laboratory if you have some interesting strand in which you are interested. So these very important discovery about fungi will lead you to play with fungi inside your laboratory, like biosynthetic gene that are responsible for metabolic pathway are encoded. That code for secondary metabolic pathway are clustered in fungal genome. So this is one of the important discovery. The whole genome sequencing has revealed that the fungal biosynthetic pathway has been unexplored. Only limited compound has been identified, while the biosynthetic pathway show some more compound which is still cryptic or orphan. And another thing, the profound knowledge about the polyketide synthesis, this one, and NRPS, non-ribosomal peptide synthesis in the hybrid. So we have profound knowledge about this. So these things can lead us to what? Can have new compound from fungal source, but if you don't have that, then another thing is that you can manipulate the media by traditional way, or you can go inside the genome to awaken the silent gene, like study how to discover the cryptic natural product, which is not discovered by normal condition, ordinary condition, like by triggering how. So you can trigger. Two methods are there. One is traditional method, that is called osmic, one strand many compound. You have one strand and you can have a lot of compound just by changing the media, just by changing the condition, environment, like how heat shock, temperature and other. So the other is co-culturing. These two are very common, but the more advanced is the genomic based approach, and that is genomic mining, epigenetic remodeling, and activation of pathway specific regulatory gene. So these are the different possibility in which you can explore the cryptic natural product. And traditional, just look into that. I will say that I will not take a lot of time with this thing, but will come to my original work, so you can just see the slide. Traditional, like changing the culture, changing the culturing condition. These are the different stress factor. Like I said, that temperature, light, media. You can grow it in the artificial seawater and deionized water. Okay, these are the example. Aspergillus ochraceus can produce, normally it have one compound, but literature shows that we have selected 15 new compound from the same species by changing the condition. So these are the references. Co-culturing. By co-culturing, the species can interact by chemical signal or physical interaction, in which it stimulates or which it can trigger the biosynthetic pathway to have new metabolites. And these are the example from literature. Genomic mining, how? A combined genomic analytical approach is a powerful tool for discovery of novel natural products. This is the more advanced way. And cell co-prediction of potato chemical structure encoded by ERPN genetic loci and enable the direct search for relevant metabolite. How? You can predict that this strand can produce such a compound, and you don't have that in normal condition. In order to get that, you can manipulate. So, for example, Aspergillus neduline. Look into the Aspergillus neduline. So, it has the possibility to produce different type of compound like quinoline, quinoline, acridine, and that's why it has get isolated 4 novel quinoline alkaloid. Well, it was predicted first and then it get isolated. Epigenetic remodeling is very complex but very interesting. You can use chemical which act as a epigenetic remodeling. So 2 targets are there: DNA methylation, so here, and histone modification. Well, for the histone modification, we have HDAC, histone deacetylase. So to inhibit this one, and histone methyltransferase. To inhibit this one, you can manipulate the metabolic profile of the fungal strand. Well, here you have DNA methylation. If you inhibit that by chemical, any compound that you use, different compounds are available. We have used a number of compounds like SAHA, suberyl analyte, and you can use different, and that way you can get a number of new compounds. So these are the references, and this is by epigenetic remodeler modifier, and how it how it inhibit the HDAC DNA methyltransferase, and how it activate the biosynthetic pathway to get the metabolite. And this is the example. Here you see that this compound inhibit the DNA methyltransferase to get this one. While in the Aspergillus neduline, this SAHA, suberyl analyte hydroxamic acid, inhibit the HDAC to get this compound. And this is the last one, that is the activation of pathway specific regulatory gene. So you can, some protein are associated with genome responsible for the biosynthetic pathway. If you activate that, you can awaken that, you can have different compound like this one. So from DNA we have transcription RNA here, that. So these are the enzyme or protein associated with this pathway, and if you activate that, this is the transcriptional factor. So if you activate that, it will produce new compound like here as a biosynthetic pathway. Now, what we have done, this is just from literature, this is as a prelude which have attracted our attention to work on the fungi. So what we have done, our laboratory procedure to have small-scale extraction, preliminary cytotoxic screening, then dereplication exercises, and then to have final complete structure of the compound and to repeat the assay whether it is active or not. So small scale extraction is like that. We grow that in the 125 ml scale like here, and this one, and this grow per 21 days on small scale to in the different media like here you see Zephyrox, maltose. Either you grow that in the deionized water or in the artificial seawater. Either you can extract the broth and mycelia together or separately. So this is small scale for the screening purposes. And once we have the crude extract from this extract, we send it to the Henryport, Michigan Cancer Center to screen it against different cancer, solid tumor cancer cell line. So look into that. We have different solid tumor cell from murine and human. And it is relative, murine solid tumor selectivity versus murine leukemia in normal cell. So you have different like lungs, colon, brain, breast, prostate, ovarian, and murine leukemia and human leukemia. Well, how can you put the top priority? If it is, if a extract is active against the human solid tumor cell, then it is put on the top priority, and then on the murine. And if it is active against the both, like the murine in human and leukemia, then that is the top priority because it's a potent one. And this activity it is the 8 mm is the normal one. So above 8 mm inhibition, so it is considered that is the solid tumor selective zone assay. You can read about and quite detailed. This is the reference by Jasper Port Cancer Brett Villaret. This one, Villaret. So here we have the result from Henryport Cancer Center. Maybe it's not quite clear, but it's the actual result, and I will show you this one. So look into this. Two candidate, this one, that is the strand code 088708, and this one 088702A. I have worked on this one while one of our friend, colleague, Dr. Wu from China worked on this one. So both the strands were collected from Hawaiian shell water sediment, and this is the dereplication exercises. What is the, first I will show that how many strands we have evaluated or screened for the activity purposes, what we selected for further work, and what we discarded. So look into that. This is the overall result, 16 fungal strands. We have dereplicated 16 fungal strands using the LCMS, UV, mass, and on DNP. And we decided not to go on 9 project because we were not expecting any good compound or new compound, and then 5 projects were selected to go on, but it didn't produce any new compound, so we stopped working on that. While the 2 projects, this one, 702 and 708, we observed some new peaks and the dereplication exercises. And this one, I already told you that Dr. Wu work on that while I work on this one. So here, though it is pooled by penicillic acid and dihydropenicillic acid, but still 2 additional peaks were of interest to us. And this is what? This is the strand which I have told you that Dr. Wu worked on that, the 088702, which were taxonomically identified as Aspergillus and Salicola, and she get isolated the novel dipeptide azonosine, and this is the structure. But I would not talk about this because I haven't worked on that, and I have worked on this one. 702, this is 088702, which is Aspergillus ochraceopetaliformis. And look into the chemical profile, HCMS profile, you will see here. These were the 2 peaks, these were the 2 small peaks, that is 489 and 477. we didn't observe any hit in the dereplication exercise on D and Phi, and that's why we decided to get isolated these two compounds. Look into that. Okay, so this was the procedure. After dereplication, we grow the fungus strain on a large scale, that is 12 later for 30 days instead of 21 days. We, in order to get enough material of that, we grow that and look into the fractionation method. Ethyl acetate extracted, then fractionate into 3 fraction, hexane fraction, and dichloromethane FD fraction, and aqueous. So we didn't work on these two because the two compound, the two peaks were present in the dichloromethane fraction. So we use the SP in order to solid phase extraction, in order to get rid of the other compound and scale up the compound, the scale of the two peaks. So look into the fraction which we have get isolated 1, 2 up to 8, but the peaks were present only in the F4 and F5. So we get combine it, and the two compound Ochrine A and B were get isolated. This is the look into this fraction. So this is the LC profile. These were the two peaks. Now it looked quite bigger because it was scale up, while this one, that is pleocol. So it is the extra compound. So if you remember the previous prochemical profile, there were a bunch of peaks, and we removed that by fractionation method, and we just concentrated the two compound here. Look at the 489 and 477. And this is the Aspergillus ochraceopetaliformis large scale growth, and these are the final structure. So it is cyclotetrapeptide, isoleucine, threonine, tyrosine, proline. The only difference is this one, isoleucine, threonine, tyrosine, but instead of proline, we have alanine and the Ochrine B. So both are quite close to each other, with the only difference of one carbon. Look into that. This is one carbon short, while this it has one carbon more. So the difference is alanine and proline. This is the physical data. There is the molecular formula, mass, yield, how much yield we have, and different other physical data. Ochrine A, we were facing serious problem of what? Solubility. We couldn't dissolve it easily. We tried a lot of solvent, like look into that limited solubility, chloroform, dimethyl, acetonitrile, and deuterated water, but we didn't solve that. At last, we dissolved the compound and the pyridine with some cation. It was not really good, but rather it was suspension, but pyridine was a little bit more good than other, and then we get that because the peptide nature were observed like in the proton NMR, if you interested in that. So this is the carbon 13. You can see that the 4 carbonyl appear here, and the other characteristic peak for the peptide nature. And this is the other one, that is Ochrine B. So this is the proton NMR spectra, and this is the carbon 13. The difficult task was not only to identify the amino acid, but the sequence. So we use systematically like that. First we acid hydrolyze, and ESI-TOPMS analysis was carried out to know what type of amino acid are present. So the first thing was that these amino acids are present in Ochrine A, the first one, proline, threonine, isoleucine, tyrosine, while in Ochrine B, alanine, threonine, isoleucine, and tyrosine. The second task was that to determine the sequence, and the sequence of the 4 amino acid and the cyclic form was determined by 2D mass spectrometry, 2D NMR, and the mass fragmentation pattern. So Cousy, HMBC, and mass fragmentation pattern will become able to develop a sequence. While the next difficult task was to assign absolute configuration, and that need a lot of that much more quantity of the two compounds. For that purpose, we have used the Marfey method, and in Marfey method you will acid hydrolyze it, and then you will derivatize with Marfey reagent, and then you will analyze it. And I think that it takes a lot of time, because you will leave the compound for 16 hours at 110 degrees C. So we come to the conclusion that in L and D, only L amino acid is present. So you have two isomeric amino acid, either L or D. But in both compounds, only L amino acid present. And then you have to determine that whether it is L simple L or L-LO. But we observed that no L-O, L-LO amino acid were present. Only L amino acid were present. And that is quite strange. In nature, you will hardly find any, because we have some example, but only few. And cyclotetrapeptide with only L amino acid, that is quite rare. And you can see for that purpose we have compared these report with these two compounds with reported one. Here you can see, and only few are here. Look into that. These 5 examples have that have same nature, like all the amino acid are L type, L isomer. So the question is that it is difficult to synthesize, or no synthesis. Are they correct? This is the question which struck my mind, and why it is so. So I questioned the presence of these Phenystein and Phenystein B. So the paper is here for reference. You can study that, but it's published quite earlier, and that was that how it is possible. And so I'm still worried about that, whether it is very rare, or can we synthesize it, or nature can produce it. But it's still that all the amino acids are L isomer. Once we completely characterize the two structure, then we have to refute that whether these are the active constituent for the selectivity of solid tumor or not. So again, we send the two compound to Jaspon Port Cancer Center to screen it against 12 cancer cell line, but no significant result. And then we send it to University of Berkeley to test it against these, and again we couldn't observe any significant result. I think that the problem was the solubility, and they didn't use pyridine for solubility activity purposes. So I think that maybe it is due to that. But still, we didn't stop there. We sent it to the University of California Screening Center for the high throughput screening, and we couldn't observe any significant result. But the thing didn't stop here. Again, I sent the compound, isolated the compound, and surprisingly, quite recently I have got the result against it is active against HC virus. But we will set a microbiologist over that whether what's the relationship, what's a structure, what is the moiety responsible for this activity, can we reproduce it or not. So this is the essay from which I said that no significant result from the Jaspon Port Cancer Center from Michigan. No result, no significant result. Look into that. So this is the University of Berkeley essay, no significant result. And the conclusion is that it seems that we are passing exciting time where the marriage of fungal nature product chemistry and molecular biology. I had told you that genomic mining enable you to play with fungal strand in your laboratory, and you will have new or novel compound that will lead us to a healthy life, lead you to a new drug discovery which will give you a healthy life. Okay, thank you very much.